day. This is the Professor, CBMD PhD, and today is Monday, July 17th, 2023. It is 2:48 and 7 seconds, AM Eastern Standard Time, and today's research, immune memory. So let's jump to it here. Immune memory is achieved by epigenetic and topological rearrangements of DNA in immune cells. Date July 11, 2023. Source: Joseph Caretas, Leukemia Research Institute. A team of researchers has discovered that the memory of the immune system relies on characteristic epigenetic features and a specific 3D arrangement of the DNA in the nucleus of memory cells. Allowing for a fast activation of these cells, these features are altered in chronic inflammation diseases like asthma and can be important in autoimmune diseases and cancer. The immune system is one of the most complex parts of our body. It keeps us healthy by getting rid of parasites, viruses, or bacteria, and by destroying damaged or cancer cells. One of its most intriguing abilities is its memory upon first contact with a foreign component called antigens in scientific jargon. Our adaptive immune system takes around 2 weeks to respond, but responses afterwards are much faster, as if the cells remembered the antigen. Researchers compared the response of immune cells that had never been in contact with an antigen, called naive cells, with cells previously exposed to antigen, memory cells, and sort of doing. They focused on the differences in the epigenetic control of the cellular machinery and the nuclear architecture of the cells, two mechanisms that can explain the quick activation pattern of memory cells. While all the cells in an individual have the same genetic information, different cell types, access to different parts of the DNA, the term epigenetics encompasses the mechanisms that dynamically control this access. The results of this research team revealed a particular epigenetic signature in memory cells, resulting in the rapid activation of a crucial set of genes compared to naive cells. These genes were much more accessible to the cellular machinery, in particular to a family of transcription factors called AP1. To put it into a racing context, these genes have been warming up ever since the cell's first contact with the antigen. However, this epigenetic signature was just the tip of the iceberg. It is known that this position of the DNA in the nucleus is not random, but reflects the cell's activation state. The researchers found that, indeed, the 3D distribution of DNA in the nucleus is different between naive and memory immune cells. The key genes for the early immune response are grouped together and under the influence of the same regulatory regions, called enhancers. Keeping with the racing metaphor, the genes are not only warmed up, but also gathered together at the starting line. Although most of the research has focused on healthy cells, the scientific team wondered whether any of the mechanisms found could, when altered, explain actual diseases in which the immune system plays an important role. And to address this question, they analyzed immune cells from chronic asthma patients and found that the circuits identified as key for an early and strong immune response were overactivated. The epigenetic control of the immune system is a blossoming field, and discoveries like the ones by researchers are setting the stage for the next generation of epigenetic drugs and treatments targeting autoimmune diseases and cancer. And this is the Professor of Microbiology, Bioinformatics, Pathophysiology, and Leader of Research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.